I'm Naomi Newsom.
I'm Whitney Butler.
And we're here to discuss an educational video about CRISPR/Cas9 technology. So, what is gene editing? So, gene editing is where the DNA sequence is altered by either insertion, deletion, or modified or replacing the DNA sequence, which in turn is causing a modification with the gene function.
Why is it important, or how does it relate to CRISPR? CRISPR Cas9 technology is a tool for gene editing, and the technology also allows to correct genetic deficient, treating and preventing diseases. What does CRISPR stand for? CRISPR stands for cluster regularly interspaced short pandemic repeats, and Cas stands for CRISPR associated. Where CRISPR originate and natural and purpose. What is this purpose? CRISPR happens in the immune system and bacteria to fight against foreign genetic elements. What does CRISPR do? CRISPR/Cas, which Cas is protein classified as an enzyme that acts like a pair of molecular scissors for DNA.
So, based on people's understanding of nucleic acids, what type of bonds form between the CRISPR guide and the RNA, the CRISPR/RNA guide molecule and the target DNA? And what type of bonds would an enzyme such as Cas9 affect? So, of course, the bonds between the target's DNA and the RNA, the guide RNA, which is gRNA, has hydrogen bonds that form between these two. The reason for this is the target DNA has a partially negative charge, and this allows the attraction between the RNA, which is a partially positive charge. So Cas9 enzyme would affect the hydrogen bond by breaking it apart. So basically it's splitting it down the middle. Whitney, would you like to show me? Boop. Okay. So, sorry guys. Okay, so we're going to compare the construction of gRNA, which is guide RNA, versus cRNA, which is CRISPR RNA. So guide RNA is used as a guide to the target DNA. So Cas9 can cleave it off. So basically it guides the Cas9 to the target, and it's able to cut it. This is the Cas9, boom, break it apart. So cRNA, which stands for CRISPR RNA, which contains spacers and partial repeats. So spacers are complementary nucleotides target, so they complement the DNA target that we already have. So the Cas protein can come in and break it down. So basically they're the same thing. They're used as guides, but one is particularly used for the CRISPR. One is particularly used for the CRISPR, and one is particularly used for the protein. So in a sense, they're both seen as guides. So, when the Cas9 is induced with cleaving and resulting in NHEG, and it causes a mutation in a cell, one common effect is a frame shift mutation. What is a frame shift mutation, and how does it affect the gene expression? So, the frame mutation is caused by deletion or insertion of a nucleotide in the DNA sequence. So this is causing a shift that affects the gene expression. If you think about translation, which transcription, translation. So transcription is DNA going to RNA, and that's where the frame shift is happening, which is causing either the protein or the gene, basically the amino acid, to either have a shorter sequence from the original one, or causing it to be longer. So this is basically from the original state. So basically that's what it is.
So HDR stands for homology, directed repair. HDR is seen as an alternately way after the cleavage of DNA.
So like breaking apart.
So that's similar to
breaking apart the DNA.
Right. So a donor construct which contains the selectable marker on one side of adjacent sequences, causing this to act template for HDR, and results in the insertion of marker into the targeted site.
So basically, the HDR removes the sequence from the NIC DNA, then the donor construct is added to reduce the risk of frame shift mutation because of only one sequence is being cleaved off instead of having two compared to NHEG. So this is more desirable because it's causing less mistakes to happen, less mutations to happen, and it's basically not foul-proof, but it's a little bit better than the other one. So
it's two types of Cas9s. So it's a dCas9 and it's a wild type Cas9. How is the dCas9 different than the wild type Cas9? Is able to repress transcription.
So basically, the Cas9 is used to cut the DNA, which leads to a gene shutoff. And dCas9 is a protein that lacks catalytic activity against DNA. So it lacks basically the type of enzyme to break down the DNA because it has point mutations. What point mutations are little additions of nucleic acids within the endonucleus, which is a type of, let's just say a type of enzyme. And this leads to dCas9 not breaking off the target DNA. But this represses transcription, which is repressing DNA being basically coded to RNA, which is basically blocking our RNA polymerase from binding and basically coding for it, elongation. So it's coding for it. So how can dCas9 be further modified to promote transcription? So dCas9 can be used to promote transcription by using the guide RNA, which is gRNA, which it can guide the insertion or deletion of uridine, which is a particular nucleic acid, to RNA, residue in two mRNAs. So it fuses, meaning it binds to the dCas9 activator to the upstream region. So this of the promoters, which is leading, so it's increasing more RNAs to be transcribed, I think. That's not how you say it, transcribed.
Yes.
It's transcribed of the gene interest. So it's promoting more of the RNA to be transcribed because it's binding to the dCas9 activator is binding to the promoting site. So I'm going to hand it off to Miss Winnie for the last part. And
So discussing an example. Okay, so I'm going to discuss an example of biotechnology. And for CRISPR technology, it can be used to create vaccinations to better fight diseases. For example, HIV/AIDS vaccination is altered. It alters the DNA sequences, making it harder for HIV/AIDS to attach to the immune system and binding to the T cells,
Which
it also creates.
Yes, which the vaccination basically is going, of course, vaccination is going to prevent HIV and AIDS. So it's going to make it, like she said, make it harder for the HIV cells, well, HIV basically cells, to attack and insert its DNA into it. So you were right. I'm sorry. I'm so sorry.
Our friendship.
Dr. Thomas. Okay. You know we're nervous. Okay.
Yeah.
This took how many times? Okay. We're going to keep this in here.
Yes, we love you, Dr. Thomas.
Really, we do. Okay. Sorry, let me go to the last one. Okay.
And it also created pest resistance crops by expressing gene that makes it hard for pests to bind to plant crops.
So basically, just think about, your regular agricultural fields for foods and stuff. It makes it harder for little pests to, get on the leaves, causing it to die and basically get, what would you say, an infection, basically?
Yeah, killing it.
Yeah, killing
Trying
it.
to kill it.
I would say this is more of a natural way compared to using chemicals. So I think this is more of a alternative way I think people are turning to for using this. I think a lot of people have used CRISPR for plants instead of humans mostly, because we're starting just to dive in this because it's fairly new. So there's not a lot of information on it too much, but there's a whole lot of information on gene editing and gene engineering. So I think, how do you think we did?
I think we did good.
Yeah, so I hope this was very educational. I hope we explained it to the best of our knowledge and iterated it enough for you guys to answer the handout that we had. On the handout, we particularly just have 5 objectives. We're having 5 questions, right?
Yes.
And an activity where we interpret the CRISPR/Cas9 technology from a research article by using a figure. Is there anything else you'd like to say, Miss Whitney?
Y'all are going to do great.